Hello friends, myself Bhavan Jha. Today I have these topics from class 11th biology. These topics are important from class exam as well as medical competitive exams. Today's topics for discussion are symmetry, coelom and segmentation from chapter 4 and photorespiration from chapter 13. First one is symmetry. Symmetry means balanced distribution of duplicate body parts or shapes within body. On the basis of symmetry, animals are three types. First one is asymmetrical animals. In such animals, any plane passes through center doesn't divide them into equal halves. Example, most sponges. Second one is radial symmetry. In radial symmetry, any plane passing through central axis divide them into two equal halves. Animals with such symmetry are called radiata. Example, cnidarians or coelenterates and ctenophores. Next symmetry is bilateral. In bilateral symmetry, only one plane passing through center divide them into two equal halves. Such animals are called bilateria. Example, platyhelminths or flatworms to chordates, that is platyhelminths, aschelminths, annelida, arthropoda, mollusca, echinodermata, hemichordata and chordata. But in echinoderms, symmetry is ARLV, that is adult radial, and larvae have bilateral symmetry. Next one is coelom or body cavity. On the basis of coelom or body cavity, animals are three types. First one is acoelomate animals. Animals without coelom. Example, flatworms or platyhelminths. Next one is pseudocoelomate. In pseudocoelomate animals, body cavity is not lined by mesoderm. Instead, mesoderm is present as scattered pouches in between ectoderm and endoderm. Example, aschelminths or nematodes. Nematodes are only pseudocoelomate animals. Next one is coelomate. In coelomate animals, body cavity is lined by mesoderm. Example, annelida to chordata. Next is segmentation. It occurs only in three phyla of animal kingdom, that is in annelida, in arthropoda and in chordata. Segmentation is mostly external in arthropoda and mostly internal in chordata. Metamerism. Body is divided externally as well as internally with a serial repetition of at least some organs. Such segments are also called metameres. It is true segmentation. It occurs only in annelida. In earthworm, there are 100 to 120 segments, whereas in leeches, there are 33 segments. In tapeworms, there is false segmentation or pseudo-metamerism occur, where proglottids or false segments are budded off from neck region. Next topic is photorespiration from chapter 13, that is photosynthesis in higher plants. Photorespiration is respiration initiated in chloroplast under high light conditions. It involves interaction of three cell organelles, that is CPM, chloroplast, peroxisome and mitochondria. In high oxygen concentration and higher temperature, rubisco, that is ribulose 1,5-bisphosphate carboxylase oxygenase, functions as oxygenase because site of this enzyme is the same for both carboxylation and oxygenation. So it leads to formation of one molecule of 3-PGA, that is 3-phosphoglyceric acid, and one molecule of phosphoglycolate, which has 2 carbon. Phosphoglycolate is converted to glycolate and transported to peroxisome. In peroxisome, it is oxidized to glyoxylate and then into glycine. Now glycine enters into mitochondria and forms serine and carbon dioxide. Serine is taken further by peroxisome and converted to glycerate by a series of reactions. Glycerate now enters chloroplast and forms 3-PGA. Now 3-PGA molecule enters Calvin cycle, and so 75% of carbon lost during oxygenation of RuBP is recovered, but 25% is lost as one molecule of carbon dioxide is released. Since there is no synthesis of sugars or ATP or NADPH, so it is considered a wasteful process. But photorespiration also has useful role in protecting plants from photooxidative damage. Photorespiration occurs only in C3 plants. It is absent in C4 plants because C4 plants have crunch anatomy. Thanks for watching.